This is a production of Cornell University.
So thanks very much. I appreciate the opportunity to speak to Horticulture today about this really bizarre event. And I think, so how many of you grow tomatoes? And yeah, and how many of you had problems in 2009? Yeah. So it was absolutely bizarre, and I thought I'd just like to explain a little bit about that. Some of the it was a pandemic. It made the national and international news. Here's a quote from the New York Times on the 17th of July, 2009. It says, "A highly contagious fungus that destroys tomato plants has quickly spread to nearly every state in the Northeast and the Mid-Atlantic, and the weather over the next week may determine whether the outbreak abates or whether tomato crops are ruined." And then in August, there was this headline that appeared in the New York Times. And according to this guy, he says, "You say tomato, I say agricultural disaster." And that was from an organic tomato grower who ships directly to high-end markets in New York City. And he lost a lot of his tomatoes that year. And I think many people had the same experience throughout the Northeast. Actually, my tomatoes weren't too bad. In 2009, I went to Empire Farm Days in August, and I was talking with some people, and the guy who lived not too far from me, a couple miles away, I live in the town of Enfield, he said, "I lost all my tomatoes." I thought, whoa, I'm going to start spraying my tomatoes. So I did, and I actually did okay in 2009. What I want to do is to give you some very introductory comments. There may be a few of you who don't know that much about late blight. If you have questions about late blight, I'm going to pass them to Brian, because I talk a lot about late blight in my class. And I'm going to talk, first of all, about migrations of the pathogen Phytophthora infestans, worldwide migrations, but also migrations that have happened within the United States. I'll focus specifically on what happened in 2009, and then what's happened subsequent to 2009, and I'll finish by giving some predictions about management of this particular disease. I want to describe to you, and actually what I should do is ask Brian to describe this. This is the asexual cycle of Phytophthora infestans, but I just wanted to illustrate it to you. This organism reproduces by producing sporangia. And this is a micrograph of a sporangium. Here it's giving birth to a zoospore. These zoospores are about 10 micrometers in diameter. And they're interesting because they have two flagella. Here's a schematic drawing of them. A forward-directed tinsel-type flagellum and a posterior-directed whiplash flagellum. And they are motile for minutes to up to an hour. If this zoospore lands on a leaf and penetrates the leaf, it can produce a very small lesion that looks like this in about 3 or possibly 4 days. It takes at least 2.5 to 3 days before you see any symptom at all. And this is probably 3 to 4 days old. Within a couple days, that lesion can expand, and the pathogen has grown throughout this tissue. And if that lesion is subjected to very moist weather, leaf wetness for 10, 11, or 12 hours, it can sporulate. Brian, how am I doing?
Pretty good so far.
Okay, thanks. All right. So this is the pathogen sporulating on the leaf. Now, Brian, how many sporangia are on that leaf?
Well, we had another comment in the audience about the type of plant that is actually. We're not quite.
Oh, okay. He wants to hear your question.
Oh. No, it's Brian. He doesn't deserve it.
All right. That goes with the name? So this is a potato leaflet. This pathogen goes to both tomatoes and potatoes. It's actually more famous, as I'll say in just a minute, as a potato pathogen than as a tomato pathogen. That is to people who've worked with it for a long time. People who grow tomatoes don't know that necessarily, but it is. Anyway, there are probably more than 1 million sporangia on that leaflet. And if you were to look at this with a dissecting scope or a hand lens, you'd see the sporangia among the trichomes on this potato leaf. If you were looking at it through a compound microscope, you'd see the sporangiophores, and here's a sporangium. When the weather changes in the morning particularly, and the dew dries off of a leaf, these sporangia are actually discharged, and here's one that has been discharged, and these can be aerially dispersed. If they land on a surface that has free moisture, they can germinate. So this is the asexual cycle. It's really dramatically important. It can happen in between 4.5 to 5 days, depending on weather, depending upon how susceptible the host crop is. And it's really quite dramatic. And it's so dramatic that it can turn a field of potatoes that looks something like this within 8 or 9 or 10 days to a field of potatoes that looks something like that. And in another 8 or 9 days, it can look something like this. So I can tell you that every last bit of potato, and this is a potato field, thank you, Brian. Every last bit of potato foliage has been destroyed. Now you'll see some green here and here, that's pigweed. And this is a grass of some sort. But all of the potato foliage was dead. I took this picture in August of 1994. This is a late season cultivar. It should have been green until mid-September, excuse me. So it was totally dead well over a month before it should have been, and that was caused by late blight, caused by Phytophthora infestans. So in addition to destroying foliage, this pathogen also infects tubers. And this is a really important fact, is that the pathogen can overwinter in infected tubers in a potato storage, or if there are tubers left in the soil and the tubers don't freeze, then it can survive in volunteers the next year, or it can survive in great big piles of cull potatoes, one of which is pictured here. And this is, I took this picture in, I think, 1994 also. And actually, Don Hall set this pictured in this, this image. Don, do you know where you are?
Steuben County.
You were in Steuben County, yeah, but I mean right here. It's really good you know where you are. So this is tons and tons of tubers that were dumped because the grower had harvested the tubers about a week previously. He'd had late blight in the field. It was really warm and wet that harvest season. Those conditions are just perfect for infection with Phytophthora infestans. So the potatoes were put into storage. They were warm, they were wet, and late blight lesions on a tuber are spectacular places for Erwinia carotivora or Pectobacterium carotivrum can infect and cause soft rot. And so these potatoes were just melting in storage. It was just going down. And it was a huge storage. John, do you remember how large that was? It was
Probably about 10,000 hundredweight.
about 10,000 hundredweight. So can you imagine, these potatoes were about 4 or 5 feet deep, all the way down into the bottom of that gully, which was 40 or 50 feet down. What I'm thinking of is the amount of bacteria, Pectobacterium, that was going downstream from this pile of potatoes. Probably most of the pathogen did not survive in this because the tubers were so rotten. I want to talk a little bit about the sexual cycle of this organism because that's also important. This is a picture of an oegonium, and here's an antheridium. So this is the female structure, this is the male structure, and a nucleus migrates from the antheridium into the oegonium and fertilizes the oegonium. And once that fertilization occurs, then we get an oospore identified here. And you can see it's a very thick-walled structure. These oospores can survive for years in soil. As far as we know, that does not yet happen in the United States. There are places in the world where it does happen. It certainly happens in central Mexico, in the highlands of central Mexico. Within the last 20 to 30 years, we know that it happens in Northern Europe, particularly in Scandinavia, and also in Northeastern Europe. But in most places in the world, and I think also throughout the United States, we don't know yet that oospores occur, because we don't think sexual reproduction is yet a factor in the United States. So most people have heard about potato late blight because of the famine it caused in Ireland in the mid-19th century. This book is actually a fascinating book. It's by a female author who took a male pseudonym to write about it. It says 1845 to 1849. It's really just a chronicle of what happened. There's not much emotion, except the facts and figures are so awful that they're just presented in a really bald way. And there were many cartoons, if you will, in newspapers. This is one from that period in which this represents Ireland. It says help, and it says we are starving. So the Irish potato famine was really awful, as many of you know. Late blight was not known to the Western world until the mid-19th century. And this is a picture of the development of this new disease in Europe starting in 1845. The first occurrence was detected in Belgium at the end of June. And then this new disease appeared later in the season as we went through the summer of 1845, until by the end of the season all of Ireland had been affected by the late blight. One of the reasons that Ireland was so severely affected is that potatoes were just a huge component of the diet for people. For example, if you were among the lowest class, the cuttiers, smallholders, and landless laborers, your calorie intake was from eating 12 pounds of potatoes a day, and this is for most of the year. And so when that food source was destroyed, you can see what it would do to one's calorie intake. What I learned, which was really fascinating, is that potatoes probably caused a population explosion in Ireland. There were no censuses in Ireland in the 18th or 19th century. But it's estimated that the population of Ireland at the end of the 18th century was about 3 million people. It's estimated that by the mid-19th century it was about 10 million people. And potatoes had become a major calorie source in the end of the 18th century in Ireland, which enabled this huge population explosion. And it wasn't uncommon for mom and dad to have 18 kids. They could subsist on potatoes. They could heat a one-room stone house with peat. And it really created this very huge population explosion. Okay, now I want to talk about migrations worldwide, but mainly in the US. The reason I said mid-19th century for Western science knowing about this disease is because it appeared first, it appeared first in the US, maybe around Philadelphia, around New York in 1843. And here's this illustrates how it radiated out from 1843 to 1844 to 1845. So one of the questions is, where did this organism come from? And I'm going to suggest to you that it came from central Mexico. There is a significant debate about where this organism came from. It was debated at the end of the 18th century and through the 20th century, or at least until the middle of the 20th century, when everyone, that is most scientists, agreed that maybe it came from central Mexico. Then in the late 20th century, some of my colleagues suggested, no, it didn't come from central Mexico, it came from South America, where potatoes also originated. I think the developing data now will confirm that it probably is originated in central Mexico. So sometime before 1842, it could have come from central Mexico to the States. It could have come from the States to Europe sometime before June of 1845. And that's easy to imagine. An infected tuber could be taken on a ship, taken to Europe. The pathogen could sporulate from that infected tuber and it could initiate disease. Or that one possibility is that it might have come directly from from central Mexico to Europe. From Central Europe, it's very clear as to how it moved because there's a very active seed potato trade from Europe to the rest of the world, except to the US and Canada. So potatoes go to South America, to Africa, to Asia, and all over from Northwest Europe. So it's easy to see how this organism could get around. In the late 1980s, there was clearly a migration from northern Mexico to the US. And the reason I know that it was from northern Mexico is that we had been sampling in Sinaloa State prior to the occurrence of new strains in both the Pacific Northwest and also the East Coast, and had found the very same genotypes of Phytophthora infestans in northwest Mexico before they arrived in the United States. So it's really clear as to where our populations came from in the late 1980s. What I want to illustrate to you is how interestingly and how fast this organism can get around. We detected this particular strain of Phytophthora infestans in the summer of 1992. It was in Oswego County. It was not in Ithaca. It was not Tompkins County. It was in Oswego County. And then we got a sample from Maine in 1993, which was also the same genotype. And it was, again, this US8 clonal lineage. In the next year, too, this same genotype was spread throughout the United States. In 1994, it was in the eastern half of the United States, and in 1995, it affected all of the western potato production areas in the United States. So this pathogen gets around in infected tubers. Those infected tubers go from the potato seed-producing areas all over. They go from, in this case, Maine, all up and down the West Coast. New York produces seed tubers, Michigan, Wisconsin, Minnesota, North Dakota, Idaho, Oregon, and Washington all produce seed tubers, and they go in trucks. So this organism gets around in trucks for intercontinental migrations. 1994, when I took those pictures, was really a severe year, and this picture was taken in August, and what I wanted, there were several reports in the newspaper about the impact of this disease on pathogens. This is a farmer, Byron Chapman, who grew potatoes near Utica, New York. He lost his entire crop in 1994 to late blight. He declared bankruptcy the next year, and he died about a year or two after that. So my conclusion was that the stress of losing his crop, losing his farm, were so severe that it cost him his life. It was all because of this strain of Phytophthora infestans. It was resistant to what had been one of the most effective fungicides, and people, when they saw late blight, would spray this fungicide, mefenoxam, expecting it to completely halt the disease, but it didn't. And when that didn't happen, they didn't know what to do. And so, and this disease is so explosive that it just destroyed these potato crops in 1994 and 1995. Okay, so what happened in 2009? So from our own Abby Seaman, we got this note on June 23rd, 2009. She said to the late blight listserv, she said, late blight has been found at several locations in our reporting area this past week. And then here's a quote from Meg McGrath on Long Island. She said, a sample just came in, there's lots of sporulation, the field had been sprayed with fungicide, and growers are taking action. And then on the next day, the 24th of June, 2009, Keith Perry, who's my next door neighbor on the third floor of Plant Science Building, came in with a tomato plant that was severely infected with late blight. He said, Bill, what do you think of this? I said, wow, it's got late blight. I said, where'd that come from? Lowe's is where it came from. So we went to Lowe's and took pictures. So these are tomato transplants that were for sale on June 24th, 2009. Not only Lowe's, but Walmart, Home Depot, and there was one more that had these same transplants for sale. And they were really all heavily infected with Phytophthora infestans. These are large lesions. You know, they have been there for a while. And so these plants had been infected for really quite some time. So on the 24th of June in 2009, we knew that Suffolk County had infected tomato plants, and we knew that Tompkins County had infected tomato plants. So here's a note from Tom Zitter on the 26th of June in 2009, and it's really fascinating. He said, yesterday I did a quick tour of garden centers in the Ithaca area. All 3 big retail stores had late blight infected tomatoes on their shelves yesterday afternoon. Some agreed to remove the plants while I was there, while others were waiting word from the New York supplier. Tomato plants have been replaced with additional plants every few days. And then he said, I also received word that late blight infected plants from large retailers were found in Cerro, Pennsylvania, Schenectady County, and as far east as Presque Isle, Maine. Presque Isle, Maine is in Aroostook County, Maine, where there's 70,000 acres of potatoes being produced. So on the 26th, these counties had reported late blight. By the 8th of July in 2009, these counties had reported late blight. So at that point, I thought, I better get out of Plant Science Building and go look around. So who knows where this is?
Oh, that looks like it's over near Fall Creek.
Yeah. You got it. So this is on Freeze Road, and that's where I went. And here's what I saw. Each of those spots is a late blight lesion. These dying, and by the way, this is a tomato plant. And these are dead and dying leaves that are infected by Phytophthora infestans. In the same garden, I looked at potatoes, and sure enough, they were infected also. And so by the 16th of June, there have been these many counties reporting late blight in New York State, and by the 17th of August, this many counties had been reporting late blight in New York State. So I thought I'd tally just do a cumulative tally of the number of reports to our diagnostic lab about late blight. And you can see that the first event was in mid-June, and here's the report on the 23rd. Here's the 24th, the 25th. And then it goes way up to above 140 reports. I want to compare that with 2008, the number of reports of late blight in New York State. So there were 3 samples received by our diagnostic lab. None of them was late blight. They were all suspected. So there was 0 late blight in New York reported to our diagnostic lab in 2008. But in 2009, we had this kind of reporting. Also thought I'd say that 37 of these 49 reports that occurred over a period of 6 days were from retail stores, which was not normal. And some of our colleagues in the state from Cornell Cooperative Extension would go to a store, find that there was late blight there, and suggest to the manager that you have to get rid of these plants, they're a problem. So what happened was that some people put them on sale to move them out. This is a picture of an organic farm not too far from here. I was there in early August 2009. They had 10 or 11 plantings of tomatoes. This is either the 10th or 11th planting. It was the only planting left. They'd removed everything else because it had been affected by late blight. These were going to be destroyed and moved very quickly because, as you can see, they are very severely affected. And that story was repeated many times in many locations. in 2009. This is a message I got from a lady growing tomatoes in Amsterdam, New York, or near Amsterdam. It's just illustrative of the impact that this epidemic had. She said, "I am an organic gardener in Amsterdam, New York, with 63 heirloom tomato plants of 23 different varieties. They're all gone. I was growing very rare varieties, blacks, greens, oranges, whites, all purchased from a very reputable grower in Schoharie. Tonight I had to leave home as my husband is pulling and bagging all 63 plants. I have 100% loss. We live on 10 acres. I inspected every day, and it seems the blight took my plants in a matter of hours. I was hoping to sell them for additional income." That always chokes me up when I read that. So that was repeated many times. So the question for us was, what was this? And what was the composition of the pathogen population in 2009? So we had received more than 70 samples. We do several types of assays with these. The samples that came from commercial potatoes were US8, the strain that we'd been seeing for really quite some time. But the ones from tomatoes were something different, and I'll illustrate that to you here. This is a protein assay that we use, and one of the reasons it's so great is we can do this assay within about 2 hours and have an answer. If we have a sporulating lesion, we can do that assay very quickly. And for example, here's the pattern that we get with US8. But in 2009, what we got was this pattern, which is rather different. Now, we also were doing DNA fingerprinting, but this assay takes us 4 to 10 weeks. We have to culture the organism, get enough DNA to actually do a southern. And this is the DNA fingerprint for the US8 strain of Phytophthora infestans. And these 4 columns represent DNA from 4 different individuals of Phytophthora infestans, which have a different pattern. And this one we named US22, because the terminology had got to that point by this time. So we had, in the last several years, we've had various strains of Phytophthora infestans. US8 is still around. US20 was dominant in 2009. And more recently, US23 and US24 are dominant. And the reason we can identify specific strains is because of asexual reproduction. So we get a clonal lineage, just all the same thing. And some of these can be very, very dominant, and so it makes sense to understand what they are. So back to 2009, we know that late blight was produced in production facilities in both New York and in Maine. At the end of the season, we had accumulated isolates from other places, and it turns out the same strain, US22, was found throughout the US. The question is, where did it come from? And here's a picture from Lowe's. Bonnie Plants has a production facility in Alabama. And it's just very clear to me that they shipped infected plants from their production facility in Alabama to the production facilities in New York and Maine. We met with representatives from Bonnie Plants in November of 2009. And this was one of their responses. "We don't ship plants, we ship plugs." Can somebody tell me what a plug is?
It's a small plant.
It's a small plant, yeah. So they've never admitted to doing anything. As far as I know, they haven't been sued, but when talks like mine happened, they got quite belligerent. But they were the cause of this huge pandemic in 2009. They have subsequently cleaned up their act quite considerably. Tom Zitter is consulting with them and illustrating how to prevent late blight. Okay, I want to move on to after 2009 and to focus on the needed IPM research that has been happening. Now, what I want to do is to focus on a tool that I think can be really quite useful. This is a decision support system which I think will enable integrated management, a very effective and efficient integrated management of late blight. It takes into account the weather, and weather is huge in terms of late blight development. It incorporates cultivar resistance, because there are some cultivars that are actually at this point immune. There's some that are quite susceptible and others that are less susceptible. There are several forecasting schemes that have been used in the US, and it contains those. It's responsive to pathogen type and also to fungicide. So it's a tool to enable a grower to take into account all of these factors. One of the most interesting things to me is the location-specific weather. You can get weather data from the nearest weather station to you. Some growers have purchased weather stations, so they have on-farm weather data. These are transmitted via telephone to actually to Bradfield Hall. And in addition to getting previous weather, the weather center here gets weather forecasts for the next 7 days, and those weather forecasts are automatically downloaded to the system. And the weather forecast can be interpolated on a 4-kilometer grid, so that you can have a weather forecast that is specific within probably 2 kilometers of you for what the weather might be. And that's really important because late blight is very responsive to weather. There are these 2 disease forecasting tools that are present in it. There's a late blight disease simulator on which one can do experiments, and I'll say a little bit more about the simulator later on. And there's an alert system that can give you information about what to expect. So let me talk just a little bit about pathogen type. I mentioned these lineages of Phytophthora infestans. In 2009, almost everything we found was US 22, and that's because it was transported on bonnie plants throughout the Northeast. And then US 8 was also present, but it was present primarily on commercial potatoes, and it gets transmitted very effectively on potato tubers. In 2010, we had a new lineage that began to appear primarily in the western part of the US. It's US 24. Also, US 23 began to appear, wasn't real dominant. US 22 was still present, and US 8 was present. And the sample size is typically between 150 and 200 for each of these bar graphs. I didn't want to do that. In 2011, US 24 became more visible, and US 23 became even more visible, whereas US 22 became less so. But in 2012, this last year, almost everything we saw was US 23. It was really a surprise. So why is it at all interesting or important to know what lineage you have or that's near you? One of the reasons is that some of these lineages are sensitive to mefenoxam, others are resistant. And here's a figure illustrating that. This is US 8, and these plates are Phytophthora infestans growing on an agar medium with either 0, 5 micrograms per mil, or 100 micrograms per mil of mefenoxam. And you can see that there is some effect of mefenoxam on the growth of US 8, but there's still significant growth of that clonal lineage on mefenoxam. But if you look at US 22, here's growth in the absence of mefenoxam at 5 micrograms per mil or at 100 micrograms per mil, and it's really severely restricted by mefenoxam. This, we put a 5-millimeter plug on the plate, so that's just about the diameter of the plug that we put on the plate. US 23 is similarly sensitive to mefenoxam, and US 24 is generally sensitive to mefenoxam. So if you have a mefenoxam sensitive strain, then you can use that fungicide, and that could be really quite effective. It also turns out that there's difference in terms of host preference. And what this slide illustrates is the amount of sporulation, that's how many sporangia are produced on a particular leaflet, for the various clonal lineages. Here's US 8, US 22, US 23, and US 24. And whether the sporulation is on potatoes or on tomatoes. And what this graph illustrates is that US 8 is actually a hell of a potato pathogen, as we already knew. But it doesn't do much at all to tomatoes. US 22 does something on potatoes, but look at what it does on tomatoes. It really is an aggressive pathogen and reproduces very rapidly on tomatoes. US 23 is even more aggressive on potatoes than is US 22, and even more aggressive on tomatoes. And then finally, US 24 is somewhat similar to US 11 in that it's a good pathogen on potato, sporulates very profusely on potato, but almost not at all on tomato. And so it really is important. Are you growing tomatoes or are you growing potatoes? Which pathogen is near you? And actually you can learn which pathogen is near you, because on the eastern part of the United States, the extension personnel have collaborated on making reports as to whether late blight is present, and they send those samples to our lab and we assay those, and we can identify to clonal lineage very quickly what that is. And for example, this is from 2012. It's a summary map of the United States from all of 2012, and essentially everything we got from the northeast part of the United States this last summer was US 23. Again, it's a really effective pathogen on both potatoes and tomatoes. What we use now are microsatellite markers to distinguish the different lineages of Phytophthora infestans. And I have data for 2011. By the end of October 2011, we had received at least 140 samples, and we'd returned around at least 80% of those within 24 to 48 hours, so that the person sending the sample had an idea of what was present. So what I think, and what we're hoping for, is to develop a strain-specific forecast that will be available on the decision support system. And I think that will enable people to become really very knowledgeable about what to do. So cultivar resistance and fungicide are both huge. And this is a picture of some field experiments looking at cultivars at Freeville. This is really a beautiful image, don't you think? Ian Small took this image. Ian has been working really hard. And I think it's really a gorgeous image. I think he was there early in the morning to go fishing, because the Fall Creek is right there, and Ian is really an avid fisherman. So I think this is even. But be that as it may, we have a fair amount of data about host resistance for both potatoes and tomatoes. This is a disease progress curve for a susceptible cultivar. And what the y-axis illustrates is the amount of disease. It goes up to 100%, which is all death. And this is time along the x-axis. And you can see in a susceptible cultivar, you get this very rapid increase in disease. For a moderately resistant cultivar, under the same weather conditions, the same inoculation conditions, you get much less disease. So that's really important. Fungicides, obviously, are really important. These vertical blue lines illustrate the dates at which a fungicide was applied. So if a fungicide is applied to a susceptible cultivar, you can suppress disease quite significantly with fungicide. But a moderately resistant cultivar doesn't need as much fungicide as a susceptible one. Or, put it the other way, a susceptible cultivar needs a lot more fungicide than does a moderately resistant one. The DSS now has cultivars by name and their category of resistance. At least 75 cultivars for potatoes, and we're in the process of doing tomatoes, but there are at least 50 cultivars of tomatoes that we can put into the DSS. In terms of fungicides, there's been also a tremendous amount of work. This is a picture from this last year. The experiment this year had at least 15 types. There were several different combinations applied to 2 different tomato cultivars and 2 different potato cultivars for something like 70 treatments, which was a tremendous amount. But we use that information to put into the DSS. And here's, you don't have to look at you don't have to understand this slide, but it illustrates how we use that information of different fungicides in terms of a disease forecast. And this is a picture of Steve McKay, who is doing the fungicide applications. And Steve was driving that tractor about 15 miles an hour between plots. The boom was bouncing all over. But Steve, it's my pleasure to recognize you today for Steve's really significant contribution in terms of all the kinds of research that goes on out at Freeville. He gets the 2011 Excellence in IPM Award. Yeah, I think anything else will be sort of anticlimactic, so let me, I'll skip this. It's really cool, but I'll skip it. Okay, so I think that with potato late blight management, I think highly localized management that's really targeted, it's like personalized medicine. It's for your farm, for that field, for that particular cultivar, will be really effective. For 2013, what we hope to do is to include sort of a targeted forecast, which also brings into account the pathogen presence or the lineage that's near you. And I think that should be able to really help out disease management. And finally, I'd like to say thanks to people in my group. Kevin Myers, who is right back there, has done all of the microsats, and people all over the East Coast know Kevin very well, at least electronically. Ian Small has been doing the work on the DSS, which is wonderful. Laura Joseph is a programmer who is just amazing. And Giovanna Danis is the person who identified the characteristics of the strains of Phytophthora infestans. And so with that, I'll quit. And if there are any questions, I'll give them to Steve. We would all love to know the answer to that question, and we're working on it. Yeah, so everybody wants to know where are these things coming from, and
makes much a difference once you got them.
Yeah, I think it was present in Florida prior to 2009, but where it came before it was in Florida, we don't know. Okay, so there's a twofold answer to that. The first is that in 2009, US 22 was spread throughout the US on infected tomato transplants. So it was just everywhere. It's not as aggressive as US 23. So we're seeing it subside in terms of its prevalence. And I think, so both things contribute. You know, widespread distribution is huge, but aggressiveness can also make a difference. And so US 23 is more aggressive. It's really going everywhere. US 22 is subsiding. What we do when we have infected plants we're not worried about, we put them into black garbage bags and let them heat up during the summer and die that way. So that's what we can do on a small scale. But as far as we know, again, knock on wood, that we don't think there's sexual reproduction yet in the United States. So I think the soil itself is not a reservoir of this pathogen. If there are infected tubers or there are other infected plants somewhere, they could be. But we think, you know, oospores in the soil are not a source of the pathogen as far as we know. Growers do a lot of things. Some growers have used flamethrowers to destroy plants. They spray a lot of fungicide. After they disc them up. What you want to do is get that plant as dead as you can as quickly as possible, so the pathogen then dies. If there are two mating types in Phytophthora infestans, if they come physically in contact, sexual reproduction is likely. Because we have clonal populations at this point, even though we have both mating types in the US, we have one clone of one mating type over there and one clone of a different mating type over here, and we don't think they interact that much. And I think that's why we haven't seen sexual recombination yet, although both mating types exist in the States. The answer is complicated to that. With the sort of multigenic resistance that we've been focusing on, we don't see any lineage-specific effect. If there's single R gene resistance, then yes, there can be quite significant differential effect. But at this point, pH2 and pH3 are resistance genes that seem to be holding up at the moment. And there are a few tomato cultivars with those resistance genes, and against this population of Phytophthora infestans, they are immune. You know, how long we'll have this population of Phytophthora infestans is anybody's guess.
This has been a production of Cornell University, on the web at cornell.edu.